Mas tengok. Hmm. Oh. Okay, get ready with your Kahoot. Sekejap saya share screen. Boleh nampak screen saya?
Boleh.
Okay, please join at www.kahoot.it. Okay, or you can use Kahoot app and enter the game pin. It's a trend ke? Kalau kita buat guna Kahoot je, you tend to use your apa? Nama samaran ke apa? Ke nama is it your nickname? Mochi, hehe. Okay. Okay, 24. Okay. All right. Okay, let's wait for your friends to join the game. 28, 29, ada yang tak stable ke? Alright, ada lagi tak? I'm expecting 6 more participants. Siapa yang ada problem untuk join? Let me know. Okay. 33. 34. Kita tunggu sampai 9:15 lah ya. Okay, then we'll start. While waiting for your friends to join the game, ada apa-apa soalan?
Doktor, any tips on final?
Saya tak dengar lah. Macam halus di hujung.
Doktor, any
Nadira,
tips for
is it
final?
you?
Yes.
Saya yang... Sekejap. Okay. Apa Nadira?
Any tips for final?
Any tips for finance? Okay. So, final exam. Okay, final exam. Okay, format for final exam questions. Will be short answer type of questions. Okay. Saya tak ada essay this time. Okay. Short answer questions. The highest mark pun ada yang 5 markah, yang lain tu standard lah, 2-3 marks. Okay. And the highest is 5 marks. So my tips is when you answer the question, you need to know how many marks that have been carried by that question. So if that question carry 5 marks, so make sure you have 5 points. Okay. If the question carries 2 marks, you need to have at least two important points. Okay. Please do not give 1 paragraph for 2 marks punya questions. Okay. Make sure give a very precise and accurate answer. Okay. Do not give 5-line sentences or 1 paragraph for 2 marks in questions. Okay, because I'm going to look for the keywords. Okay, keywords for that question. Okay.
Doktor,
Yep.
untuk submission slide, wakil atau semua kena submit, Doktor?
Wakil saja dalam grup. Okay, saya nak tanya, grup yang you untuk SCL hari ni, you all ikut grup lab ataupun you form a new group?
New group, Doktor.
Okay, new group. Okay, please submit. Okay, because make sure dalam slides tu you letak semua nama ahli kumpulan, because we have to go through your slides dalam Putra Plus nanti. Okay, because it's a really new group, so marking needs to be carried out for the whole team lah.
Ya, doktor nanti tak ada animation.
Tak ada animation? What do you mean by animation?
Kan
Maksudnya?
nanti nak present dekat projector kan. So nanti doktor ke yang kawal slide?
You all lah kawal slide.
Okay, kalau kita orang kawal slide, kita orang hantar tapi kita orang pakai yang ada animation punya lah. Doktor nak PDF ke PowerPoint?
Mana-mana pun boleh. Okay.
Oh, okay.
Alright.
Doktor, one more question.
Yep.
For final exam, topik apa? Lepas tu ada ke kena melukis?
Okay, kena melukis ada satu soalan, but it's a very easy peasy questions. Kalau you can spot anything, okay, but it's a very easy peasy questions. Okay, it's just as whether you can apply whatever. I'm sure you understand the process. Either you can apply it, okay, in the form of diagram. Okay, ada satu soalan untuk lukis. Alright, soalan yang topik covered for final exam, obviously semua topik saya. Okay, from DNA metabolism to gene expression. Okay. Ramai yang tak stable It goes down to 25 now.
Ramai tak stable doktor.
You all dekat mana? Dekat college.
Internet 2 bar je doktor.
Sedih kan? Kita ni going to online, tak tahulah, internet kita pun masih tak boleh nak support kita punya learning kan. Okay, they ask us, okay, they suggest for us to use all this interactive blah 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 blah, but still we don't have enough support, okay, especially from IT part kan. Internet, in fact dalam UPM pun ni jugalah. Ini baru saya buat online macam ni. Kalau buat dalam lecture hall lagi internet. Okay, biasanya tak berjaya saya buat dalam kelas. Okay, kalau dulu mesti macam-macam masalah. At the end of the day tinggal berapa orang je.
True doktor, internet slow.
Aduh. Itulah. Complain lah memang selalu dibuat, but I don't know, budget ke apa ke kan. No budget, no. We are looking for better facilities lah. Alright. Okay lah, boleh tak kita just continue? For those yang tak boleh nak join, you take note on the questions, okay? Okay, we can have a discussion along the way. Alright? Okay, you guys ready?
Ready doktor.
Okay. Alright. First question. Easy peasy one. Okay. So, what happened to the? Okay. When we look at these questions, sebenarnya dia give you some tricky keywords. Okay. RNA polymerase in prokaryote has a removal. Okay, what does it mean by removal here? Okay. So please be careful. Make sure you understand the questions. Okay, so do you understand why is the answer is sigma subunit? What you have in mind for those yang jawab rho factor, beta subunit? Is it rho factor is associated with RNA polymerase? Everyone? What is rho factor?
It's the raw protein.
Okay, it's a raw protein. Yeah, so it is involved in termination process. So, okay, tak ada kena mengena dengan RNA polymerase. Okay, so raw factor, it's a big no. Okay, how about alpha and beta subunit? Okay, so alpha and beta subunit is part of the core of RNA polymerase, but this question looking for the removal part. So what does it mean by removal part? Okay, of sigma subunit. Okay. Okay, the next question. Okay. It's a tricky question, but it's easy.
Saya dah tekan awal.
Okay, so do you understand why is it the answer both DNA and RNA polymerase? Because most of the answers are RNA polymerase. So saya kata tricky. Okay. Any questions? All right. Okay, next. Okay. Ini pun easy peasy juga. Okay, so it's a Tata box. So this is a promoter region. So kat mana location for Tata box? Anyone can give us? Okay, so it's a promoter. So in terms of the sequence of Tata box, it is located at minus berapa? Minus 10. Okay, so you need to know the specific location. So minus 10, so it has this Tata box. How about minus 35? Do you know the sequence of the common sequence for minus 35 region? Itu kena tahu juga. Alright. Okay. Alright, next questions. Easy peasy, but tricky. Be careful. Okay, that's why I mentioned that this is a tricky question. Okay, transcription begins. Okay, most of you answered at the start of the start codon. So why is it the answer is upstream of the start codon? Kat mana transcription tu bermula? The binding of RNA polymerase, yeah, at the promoter. So where is the promoter located at? So it's upstream of the start codon. Okay, I hope you get something. Okay. Okay, next. Okay, easy peasy one. Okay, class, most of you answered stop codon. Okay, we are discuss. Okay, this question asks you about the transcription, termination of transcription process. So basically, there are two ways. Okay, one is by protein based, or another one is RNA based or sequence based. Okay, so for protein based, we have to depend on the raw protein. So kalau for transcription, okay, tak ada kena mengenai dengan stop codon. Okay, we need to have stop codon, but for the transcription process, to stop the transcription process, there are two ways. Okay, raw protein, or by sequence base. Okay, by the sequence itself. Okay. All right. Next questions. Easy peasy. Sepatutnya semua betul kat sini. Saya baru mention je tadi. Okay. Alright, okay, most of you answered it correctly. So the upstream region which are essential for correct transcription are called promoter. So promoter is the important region, okay, to start the transcription process. So this is where RNA polymerase binds, okay, to start the transcription. Okay, ni yang jawab transcription factors ni saja-saja ke apa? Okay, next one. Okay, I'm sure you know about 5' capping. So do you know what happened during the capping process? Okay, okay. Some of you answered correctly. So, okay, we have three process for the post transcriptional. Okay, one is 5' capping. Second one, we have polyadenylation signal, and another one is splicing. So polyadenylation signal, we know that it's addition of adenine. Okay, poly of adenine. Okay. But for capping, so basically it involves its addition of guanosine, okay, GDP, in the form of structure of GDP, okay? All right. Okay, true or false? So this one for the eukaryote transcription. Okay, it's true. Okay, the answer is true. So we have Tata binding protein, TBP, is a subunit of the F transcription factor 2D. So this is the first step occurs in the for the initiation of transcription for the formation of pre-initiation complex in eukaryotic system. Okay, another true or false? Okay, senang je ni. So saya mengharapkan 100% jawab. Betul? Okay, so yeah, RNA polymerase 2, it has 6 transcription factors. Okay. All right. So next, another true or false. Oh, ada typo sikit kat situ. Okay, so hairpin loop. So it's a what transcriptional determination. What is the answer? For this, your answer false. So what is the answer? Row independent transcriptional termination. Okay, so this is a sequence-based termination. All right. Okay, another one. True or false? So what is exon?
Terkesalah.
So exon is a coding sequence. So the non-coding sequence would be introns. Okay. Yes. mRNA start codon that initiates translation. Okay, so this is you punya AUG or ATG. So this one, it asks you mRNA start codon. So it would be AUG. Okay. Non-coding DNA strand. So this is antisense strand for non-coding DNA strand. Okay. Okay, true or false? How many zinc molecule, okay, contained in RNA polymerase? So how many zinc molecule?
I think
Two.
Two, correct. Okay, so in RNA polymerase, so it has two zinc ion, all right? Okay. Okay, so this question asks you about the assembly for the pre-initiation complex. Okay, most of you answered correctly. So this is the sequence, okay, for the formation of pre-initiation complex. Okay, so these are the transcription factors required, okay, in the transcription of eukaryotic. Okay. Okay. Okay, now we move to leg operon. So like operon obviously is an example of both positive and negative regulation, okay? In either in E. coli, obviously in E. coli, okay? Okay, next questions in use of the like operon. Easy peasy. So the answer is allolactose. So do not get yourself confused. What is the role of lactose? Okay, what is the role of glucose? Okay, we have discussed on the regulation of the lag operon in the presence of lactose and in the presence of glucose. So, but now the questions ask you about the inducer. So you need to know what is the role of allolactose in the lag operon. Okay. Okay. Sequence of the structural genes in the lag operon. So you need to know the sequence, okay, in the lag operon. Okay, so this is the sequence, okay, of the structural genes in the lag operon. So we have like Z, like Y, and also like A. Okay, so you need to know what are the genes that will be transcribed from this lag Z, from lag Y, and also lag A. Okay. Okay, read the questions carefully. Okay, most of you answered correctly. So you need to know what happened when the situation has more lactose than glucose. So what happened next? So it will bind to suppressor, or it will bind. So is it RNA polymerase can bind to promoter? So you need to know the mechanism that will lead to the turn that will lead to the operation of this lag operon. Okay. Okay, easy peasy one. Lag A. Okay, so the answer is transacetylase. So this lag A code for transacetylase, lag Y code for permease, and lag Z code for beta galactosidase. Okay. Next one. Senang je ni. So, okay. Most of you, okay, answered correctly. It's a repressor. Okay, true or false? Okay, so the answer is true. Okay. Okay. Okay, you know the answer. So you understand what happened, okay, when bacterial cells prefer to use glucose when both glucose and lactose present. So in this case, we'll activate or deactivate the CAMP levels. Okay. Okay. Easy peasy one. Okay, so DNA helicase. So what is the role of helicase here? Okay, how do you explain? Okay, helicase has the ability to break hydrogen bonds. So you can understand what this means by the ability to break hydrogen bonds by understanding the role of helicase. Okay, helicase is the one that unwind the double-stranded DNA. So the unwinding of the double-stranded DNA involve the breakdown of the hydrogen bonds. Okay. Okay, another true or false questions. Simple. Okay, almost sama jawapan. So why is it false?
Prokaryotes only have one origin.
Yeah, okay, for prokaryotes only have one origins of replication, but eukaryotes, yeah, it do have multiple origins of replications. Okay, be careful, kena read the questions carefully. All right, okay. Another true or false? Okay, this one pun tricky. So the answer is false. So, so what is the answer?
RNA molecules.
Yeah, okay, you can give that answer, you can have that answer, but DNA polymerase is the one that is responsible for the construction of DNA molecules, or RNA polymerase is responsible for construction of mRNA, okay, RNA molecules. Okay, we can have both answers. All right. Okay, another true or false? Senang je ni. Okay, so it's true. Okay, primase is the enzyme that add RNA primers. Okay, kalau primase mestilah dengan primer. So straightforward. Okay. Okay, read the questions carefully. Okay, so what is the answer here?
Discontinuously.
Okay, discontinuously. Okay, so leading strand that proceeds continuously, but for lagging strand, it needs several primers. Okay, to add nucleotide, to add for the formation of Okazaki fragments. Okay, so it is processed discontinuously because they need to add they need to have more primers to be added. Okay, to add more nucleotides. Okay, for the formation of Okazaki fragments. Okay. Easy peasy one. Saya harap tak ada yang salah. Okay. Okay, most of you answered viruses. Okay, why do you answer virus? Okay, I think you can do more. You need to do more reading on virus and what type of mutations that can be caused by a virus. Okay. So the answer is splicing. Okay, yang lain tu replication, translation, transcription. So basically, is a general, it's a process. It's part of the central dogma. So it's a very important that will be carried out in both prokaryotes and eukaryotes. So splicing is basically is a mechanism that occurs in eukaryotes. Okay. Okay. True or false? Lagi 10 minit tak sempat habis. Okay, the answer is true. Okay, try to understand the questions. Okay, try to understand the statement. So you need to know what is telomerase. You need to know the role of telomerase to overcome the problem at the end of the replication. Okay, so so that you can understand the statement here. Okay, if you understand the process of what happened, okay, how the telomerase helps in overcome, okay, the replication at the end of the strand. Okay. Okay. Is it semi-conservative and discontinuous or semi-conservative and semi-discontinuous? So it is important to understand the term. Okay, what is conservative replication? What does it mean by dispersive replication? What is semi-conservative? Okay, and also what does it mean by semi-discontinuous? Okay. Okay, next one. All right, so DNA polymerase synthesizes DNA. Obviously, you have to look at the keyword is DNA polymerase. Obviously, you synthesize DNA. Okay, so kalau for those yang jawab mRNA, okay, is obviously strong, okay, because we are talking about DNA polymerase. Okay. Okay. Ask you the complementary mRNA. Okay. Nice. Nice try, everyone. So most of you answered correctly. Okay. So do you know how to read, okay, mRNA, how to get the complementary sequence for whatever mRNA or DNA that have been given to you. Okay. Okay, true or false? Okay, senang je. Okay, so I hope by having these questions, you should now able to describe. So what happened during the initiation part, so what happened during the elongation, and what happened during the termination. So that's basically covered, okay, the whole process of transcription for prokaryotic system. Okay. Okay. True or false? Okay, the answer is true. Like I is a regulatory gene. So what is the keyword here? Is a regulatory. Okay. Okay. Do you understand these questions? Who can explain what these questions ask you?
It's kind of like lactose needs to be broken down.
Yeah.
in
Okay.
compared to
Yeah.
glucose.
Okay. Let's look at the lac operon. So basically, these questions refers to lac operon, to the structural genes in the lac operon. Okay. So which are the two enzymes that are able to metabolize lactose in the structural genes of lac operon? So we have three structural genes in lac operon. We have beta-galactosidase, we have permease, and also we have transacetylase. And only two enzymes, okay, able to metabolize lactose. So can you figure out which enzymes?
The other two, like permease, is to transport lactose.
Basically, for these questions, the answer would be beta-galactosidase, obviously, to break down, and also permease, because the whole process metabolize lactose, meaning that breakdown and also to transport the lactose to, yeah, okay. So that's why the answer for this question is true. And the two enzymes are permease and also beta-galactosidase. Okay. Okay, let's move to the next question. Sikit lagi. Tak sempat tak apalah, mungkin sampai 40 ke. Okay, so tapi saya tak adalah soalan MCQ kan. If questions like this, so make sure you choose the most correct answers kan. Okay, even though ada lactose is present dekat sini, and the answer is correct, lactose is present, but we have the most accurate one. Okay, lactose present and glucose is absent. Okay. Is it true? Okay. So what happened here? Anyone? For those yang answer true, can you explain? Okay, can you take note on these questions and go back, okay, and check. Kita tak ada masa. I try to finish up to 40 questions. It's already 10 o'clock. Okay, easy peasy. Okay, so I hope you already know, okay, the location for the consensus sequence. Okay, what does it mean by consensus sequence? So you need to know the location. You need to know what are the sequence for -10 and what are the common sequence for the -35. Okay, so it's already 10 o'clock. I think I have to stop here. You all jangan leave lagi, saya belum ambil attendance. I do hope this activity do help you, okay, at least to trigger your, I hope these questions can help you to trigger your understanding, to trigger whatever that you think you haven't covered yet, okay, for the revision for final exam. If you have any questions, do let me know. Apa lagi? Kejap asyik teruskan. Kalau saya nak, macam mana saya nak? Okay, gallery. Saya ambil attendance sekejap. So I'll see you this afternoon, 2 o'clock sharp. Okay? All right. Thank you everyone. Ada soalan lagi ke? You all ada kelas ke lepas ni?
Tak ada, doktor.
Oh, tak ada. Okay. All right. No hadiah ke doktor? Oh, okay. But we don't have kita belum habis. Kita belum ada winner. Kan? We have another 10 questions to go. So how to announce the winner? You all ada kelas tak?
No, doktor.
You all nak continue for the another 10 questions? Kita skip cepat-cepat, boleh ke? Kalau you all okay, saya okay je. Yes. Okay, for those yang nak stay, so you can stay. Okay, let's continue. Okay, sikit lagi. 10 questions. Nanti kita announce winner ya, Danu. Okay. Yang ini macam saya dah tanya dah tadi. Oh, tadi the other way round. Okay. Okay, you can see that this one information we can play around with different style of questions. Okay, sedangkan soalan ni dah ditanya tadi, but different style of questions. Okay, tadi tentang 80 rich sequence. So what are they? Okay, and different style we can come up with this kind of questions about what are the sequence for them understand. Okay. See, another style of questions. Alright, ada juga yang salah. Okay, true or false? Okay, so basically these questions ask you about what are the co-RNA polymerase. So what is holoenzyme of RNA polymerase? So you need to know the what are the regions, okay, what RNA polymerase consists of, okay. So it has 2 alpha, 1 beta prime, and also omega units. I'm not sure whether I'm discuss about this point dalam lecture ke tidak. Okay, so the answer is 5. Tak apalah, this one assume extra information for you. Okay, mungkin saya tak discuss dalam kelas. Okay, on the sigma factor, on the structure of sigma factor itself. So it has 5 domains basically. Okay, there are 5 domains, so this is the main. Okay, the main domain for sigma factor. So the answer is sigma 70. If you do your own reading, okay, internet ke Google ke textbook ke, so you will find about sigma 70. Okay. All right, another 4 questions. So true or false? Proofreading function. So the answer is false. Why is it false?
Only transcribe.
Sorry?
Dia transcribe je kan.
Yeah, so
Reading ada another enzyme.
DNA polymerase, the one that has proofreading function, okay? Not RNA polymerase, okay? Okay, the last 3 questions. Okay, the answer is true. Okay, be careful. So you need to understand the description. What does it mean by semi-conservative, conservative, and also dispersive replication. Okay, so this question tells you about each individual strand is a patchwork of original and also new DNA. Okay, so you should be able to differentiate between dispersive, semi-conservative, and also conservative. For semi-conservative, you need to understand about the experiment carried out by Messelson and staff. Okay. Okay. The last two. Okay. Another style of questions. Okay, ramai yang jawab true. Kenapa false? If you remember, kita ada satu soalan tadi.
80.
80 phrase back.
Okay. Alright, nampak tak? It's a different style of is how kita pusing-pusing soalan. Alright, so why do you think the replication origins are rich in 80 base pairs? What is the advantages of having 80 rich bases for replication origins?
Weaker bonds, so can unwind easily.
Okay, so that's the answer. So you need to understand. Okay, well, okay, if the fact, okay, that origins has rich in 80 bases, there must be a reason behind that. Okay, there must be a mechanism behind that. It tells you that 80 rich bases, it has weaker bonds because it has okay, it is contain two hydrogen bonds compared to GC. Okay, GC has three hydrogen bonds, so it much collectively it's much stronger compared to 80 rich bases. Okay. Okay, final one, true or false? Okay, all right. So most of you answered correctly. So that's the end of the activity for today. So all the best. So I'll see you this afternoon. I do hope this activity helps you, helps for your revision. Okay, thank you very much, everyone. Okay, so I'll see you 2 o'clock. So, sekejap kita belum tengok winner lah.